Hi everyone. Let's go back to your digital notebook. I hope you've had a chance to fill out the chart on the second page using the Discovery Ed collage board. And you've had a chance to explore prokaryotic cells a little bit more and then compare those to eukaryotes, which we've been talking about, those plant and animal cells. So let's look at this page. It should be the third page in your notebook. And we've got a good picture here of a prokaryotic cell and some parts of that cell. And I've actually put some descriptions. I tried to place them on top of that cell part. So you could make a copy of this slide if you want to and fill out one and leave one like this so that you could use it as a study tool maybe. Remember, your notebook is up to you, so do what you would like to do with your notebook. But what I'm going to do is I'm going to talk about each one of these parts and just drag it down to its name. So I'll start with our prokaryote here. I've got this description, the region in the cell where you can find the concentration of DNA. So if you look at this cell, it's actually in here in the middle. Looks like a bunch of squiggly lines. It's a chromosome, so DNA that's been coiled up, and you could take that chromosome out of this prokaryotic cell and stretch it out, and it would be one circular chromosome. That in a eukaryote, right? We said the DNA was in the nucleus, so it was surrounded by a membrane, but not in this case, not with prokaryotic cells. So prokaryotic cells are a lot less complex. They don't have those membrane-bound organelles. And now we see that here with the DNA. The DNA is just free-floating, but it's coiled up in a region we call the nucleoid. So if you see nucleoid, don't get confused. Oops. Don't get confused. It's not the nucleus. It doesn't have a membrane around it. It's just the region where you can find the DNA, the one circular chromosome that's in prokaryotes. Okay, so also related to DNA, up here, this is about these little squiggly, they're also red, they're little squiggly lines it looks like. Those are also small circular pieces of DNA, and they are called plasmids. So they're free-floating out in the cytoplasm. They will become very important when we talk about biotechnology. Bacteria will take plasmids up from the environment, and it's an easy-ish way for scientists to get genes that don't belong to the bacteria inside of the bacteria. So plasmids are small pieces of DNA that are just free-floating out there in the plasma membrane, or in the cytoplasm, excuse me. So next, let's talk about the surrounding of this cell and every cell. So no matter what, if it's a cell, it has a cell membrane or a plasma membrane, and that plasma membrane is selectively permeable. It regulates what enters and helps regulate what enters and leaves the cell, and prokaryotes have those too. Bacteria have those as well. Next, on the outside of the plasma membrane, prokaryotes, bacteria cells, also have a cell wall. And that cell wall is important to help maintain shape, just like the plant cell, but also to keep that prokaryote or keep that bacteria from bursting when it takes up too much water or water itself. So think about multicellular organisms. Those are multicellular organisms are always eukaryotic cells. Prokaryotes are always unicellular. They are always just one organism, one cell. So that cell wall is important to bacteria to help keep its shape and keep it from bursting. Last here, this tail-like structure on the bacteria or the prokaryote is called a flagella. So that's probably pretty obvious. That's for movement or locomotion. And bacteria could use that to move toward nutrients. Some might need to move towards light. Some might use that to move away from toxins. So that's actually an adaptation, a characteristic that helps it survive in its environment for that bacteria to move. Okay, so make sure you've got that in your notebook and move to the next page where you are going to look at a eukaryotic cell and a prokaryotic cell. I want you to find things that are similar and things that are different and type your answer in that box. After you've done that, I want you to move down to this Venn diagram and drag these words where they belong on the Venn diagram comparing prokaryotes and eukaryotes. So I'm going to have you pause the video, do those two things, and then come back and we will fill out this Venn diagram together. Okay, so pause your video. Okay, welcome back. So you should have had a chance to fill out this slide. You've typed your answer here. And then we're going to go over the Venn diagram. So if a good idea is to maybe to make a copy of this Venn diagram so you can drag the pieces back up and put them in the right place just to check yourself and study. So getting started, the plasma membrane, I always say if it's a cell, it has a membrane. It's got something around it. So plasma membrane should go in the middle there of our prokaryotic and eukaryotic cells. Nucleus is a membrane-bound organelle. So that is one of those things that makes prokaryotes and eukaryotes different. So membrane-bound organelles only found in eukaryotes. So that includes nucleus. That includes mitochondria. That includes chloroplast. Okay, so all three of those are membrane-bound organelles. So they're going to be only in eukaryotes. The one that sometimes trips students up is ribosomes. Ribosomes aren't membrane-bound organelles. They're not really organelles. They come together when proteins are made. And if you remember back to when we talked about the nucleus, DNA inside of the nucleus are instructions for making proteins. Prokaryotes and eukaryotes have DNA, so they're both going to need to make proteins. So ribosomes would be found in both. Plant and animal cells up here. Actually, let me go to free-floating DNA. So DNA is in our nucleus for eukaryotes. Excuse me. Free-floating DNA would be over here in our prokaryotes. No nucleus, but it's free-floating out in the cytoplasm. Okay, plant and animal cells. So I hope you saw in the resource that plant and animal cells are eukaryotes. So that means our other organism, bacteria, would be prokaryotes. Larger and smaller, so just relative size to one another. We saw in the very first slide in Nearpod that eukaryotes were much, much larger. Remember the cheek cell compared to the bacteria. Prokaryotes smaller. More complex, definitely over here with eukaryotes, right? They have all of these organelles. They're much larger, so we would describe them as more complex. And then cell wall. We can put cell wall in the middle because even though not all eukaryotes have cell walls, they could have a cell wall. And then, of course, bacteria have cell walls. So we would put cell wall in the middle there. So check yourself, make sure you've moved yours if you need to move them in your Venn diagram. You can always use this to study, make a copy of it, use it to study. And then the last two slides in Nearpod, you have a quiz just to check yourself to make sure that you've got that material, or this material, I should say. And then your assignment is in Nearpod, and you can use this last slide here for your assignment. And when you turn in this lesson in Canvas, you'll turn in your notebook. So quick tutorial here, share. You can grab that link, copy that link, and then you can put that in Canvas to give me access to your notebook so I can check what you've done for this lesson. Okay,
and give you some feedback on that. As always, reach out if you have questions, and I'll talk to you guys the next time.